splice sites sequences, then we can apply these trained filters on the top of it, and we can generate feature maps. Each row corresponding to each one sequence, and row wise we will get feature values, right? So we can take average. There is one option to decide the threshold. We can take average value of each feature map, and the values where is greater than the threshold, the average value, then when we pick those positions and we come back to the sequence at the same position, we will point it from there up and down some length, okay, x length pattern we extracted like this. So we extracted 3, suppose here pointed, where the feature map value at this index is greater than the threshold value, and we will go there, and up 3 characters, down 3 characters we extracted. So 7 length patterns we extracted. Why we specified here 7 length only means here kernel size is 7. With that reference we extracted 7 length patterns, and we calculated frequency of all extracted patterns. The patterns which have highest frequency, for each filter we will get such patterns. For 32 filters we will get such 32 extracted patterns bunches. So from that we can extract we can calculate frequency of all patterns, and after that frequency, the patterns which are highest frequency and existing in all that filters, we extracted those patterns, and we decided those patterns as features, okay, those patterns as features which will classify the sequences as true and false splice sites. So here the strategy is given here. I'll share the PPT, please go through it. So we can extract a learned filter weights, generate feature maps, set threshold, select positions, and select patterns, store the selected patterns, and calculate frequency, extract top 5 patterns of 32 filters. Okay, based on the frequency. There are again 2 strategies. Based on first strategy, top 5 patterns from each filter, total 5 into 32. So 160 patterns are there. So out of 150, some patterns are very common in all 32 filters. So based on that, we will decide this is the pattern, and we also validated those patterns with existing patterns. Okay, with existing patterns. So like that, we will decide which patterns are crucial, are working like features. Okay, working like features. So we will do. There are other strategies to identify such patterns. If you observe this pattern, the frequency is 64. Repeated, highest frequency is 64, repeated in 31 filters. Okay, this pattern plays a crucial role, definitely. Okay, this pattern plays a crucial role, and so on. This is one type of problem. Based on that, we can classify the true and false splice sites, and we can do explainability, which patterns are working as features. So next application is we can use computational techniques. So suppose there are 7 coronavirus sequences are there. So if you go to GCID, you will get 7 coronavirus sequences. So these 7 coronavirus sequences will collect it based on country wise. Okay, based on country wise. Suppose I want to find, is there any relation between previous coronavirus sequence and current coronavirus sequence? Maybe you studied this point somewhere. The current coronavirus is divergent one compared with previous coronaviruses. How to decide that? Okay, how to decide that? So huge number of sequences are collected, and we will build a computational model to compare the sequences. There is a brute force technique. We can compare index by index characters, how many variations are there, but it's a time consuming process, and we use other computational methods or tools to do this task, sequence alignment task. There are so many tools in bioinformatics, BLAST, FASTA, and so many Mega BLAST, so many other BLAST mechanisms are there to compare the sequence alignment. Based on the output, how many sequences are similar, we can bring into one cluster, the same similar sequences bring into another cluster, and so on. There is one mega tool. By using that mega tool, so if you send the 7 coronaviruses, this is the output of 7 coronaviruses. From this, easily we can understand one point. So this NL63229E came into one branch, HKU1 and OC43 came into another branch. These 4 are human coronaviruses, means directly it will affect to humans. Next coronaviruses like SARS-CoV, MERS-CoV, SARS-CoV-2. Recent one is SARS-CoV-2. These 3, SARS-CoV, MERS-CoV, and SARS-CoV-2. What is SARS means severe acute respiratory syndrome coronavirus. Okay, 2. So these 3 are zoonotic coronaviruses. What we can say, directly humans it will not affect, through animals it will affect to humans. That's why we can call it as zoonotic coronaviruses. What we analyze from this this is a phylogenetic tree actually. So from phylogenetic tree, what we can analyze is from which genes, which new genes are generating, okay, or which new genomes are generating. So evolutionary relationship between the living organisms we will identify. What we can identify from this phylogenetic tree is the SARS-CoV-2 recent coronavirus is divergent, and it is not evolved from the previous one. It is a divergent one. With this, what we can tell to the society means, so the drugs which are worked for the previous coronavirus will not work for the current coronavirus. That type of analysis we can do it. Okay. Now, next, mutation rates. If you think about mutation rates, so the same 7 coronaviruses, if you take it, what is the mutation rate? Which type of mutations are occurring? Okay. For this, there is a tool called, just give me 1 second. There is a tool I'll share with you, that name codon suit. There is a package. That codon suit, if you pass the sequence, it will divide the sequence into codons. Okay, it will divide the sequence into codons. Frequency of the sequence, frequency it will give. Okay, so where the data science is involved here, if you think, to design such tools or to design such techniques, we use a data science or machine learning techniques. So it will give the codon frequency and so on. Okay, so if you take one reference genome, healthy genome, and the coronavirus sequence, if you take it, and which type of mutations occurred, from this table we can analyze that. So in new coronavirus, there are very few silent mutations. If you think about MERS and NL63, there are a huge number of silent mutations. Silent mutations means protein will not change. So protein will not change. If protein will not change, there is no issue. If protein will change, missense mutations, if you observe missense mutations, there are huge number of missense mutations we observed in SARS-CoV. So protein is changing rapidly, due to which, so obviously the body working mechanism, the protein working mechanism will change, it will affect last. So, and we identified there are no nonsense mutations in the 2019 nCoV. So this type of analysis we can do by using statistical methods. By using statistical methods, we will identify all these type of analysis, due to which, and we identify there are less number of silent and nonsense mutations in SARS-CoV-2, but there are huge number of missense mutations, due to which 2019 nCoV spread a lot. Okay, this is one analysis we can analyze, and different way also we can do it. Next one is translation rate. So these are different applications I'm talking. So translation rate of coronavirus, recent coronavirus. Translation rate means, at the beginning I told one, that's the transcription, splicing, and translation rate. So the mRNA or matured messenger mRNA converts into amino, what we can say, protein sequences or protein sequences. So the translation rate. So how the new coronavirus translation rate is different from exist one. There are different mechanisms or different influencing factors are there. Codon usage bias, relative synonymous codon usage, next proportions of slow codons and slow dicodons, and effective number of codons, codon position specific frequencies. By using these parameters, what we identified, this, if you think about slow codons or slow dicodons, slow codon means, see, at the translation process, give me a second. Suppose there is a, so this is my codon, first codon, second codon, third codon, fourth codon, and so on. Okay, AU, AUC, like that, the codons are there. So in translation process, biologically, so there are ribosomal subunits. It will, those two ribosomal subunits comes together. This mRNA goes through those two ribosomal subunits. Suppose this is my one codon, AUG is there. So AUG is the start codon, right? Where the translation process starts means wherever the AUG codon starts. So one more is there, transfer RNA. One more RNA is there, transfer RNA. This transfer RNA brings the anticodon and a corresponding amino acid. Okay, amino acid, it will bring it. So this is AUG. AUG anticodon, it is matched here. Then immediately it will send this AUG front and it will release the corresponding methionine amino acid, and so on. Next codon, and so on. What is the meaning of slow codons? Means the codons for which the tRNA does not exist, such codons we can say slow codons. So if slow codons are there, if there are more number of slow codons in DNA sequence, what will happen? Obviously, translation speed becomes very slow. Okay, translation speed becomes very slow. So if it becomes very slow, suppose some virus is affected or virus is mutated, the translation speed is very slow. The proteins are not generating fastly. Obviously, the spread of the virus becomes slow. Okay, so with respect to this, we analyzed the slow codons and slow dicodons with respect to all coronaviruses. What we analyzed, what we observed, and other researchers also observed is the slow codons are very less in new coronavirus. Okay, new coronavirus. So how we extracted different codes like Python programming, we used it, and some existing tools we used it. We calculated frequency of slow codons. There are some slow codons. We calculated the frequency of slow codons. We calculated the frequency of dicodons. Based on that, we decided that there are less number of slow codons in SARS-CoV-2, means 2019 SARS-CoV. So recent coronavirus have very less number of slow codons. It means there are more number of fast codons, due to which the translation speed is very high and the protein generation speed became very high. Obviously, the virus spread is very high. This type of analysis also we can do by using genome sequences. Next, if you go to sorry, I'm going little fast. I know I'm going little fast. So I'm introducing some applications where we can work in genomics by using machine learning or deep learning or data science applications. Because in the given short span, I need to cover more applications. That's why I'm going little fast. Now go to drug discovery and design. So what is the drug discovery? What drug you can do, first of all? Okay, the antiviral drugs stall the translation process, means, see, the antiviral drugs stop the translation process, means it will stop the generating of proteins. That's it. Proteins means affected proteins, like viral affected proteins, bacterial affected proteins. It will stop the translation process. That's it. Due to which thereby stopping the protein synthesis of the virus and ends its replication. Okay, so this is the main purpose of antiviral drugs. Now, how can we generate drugs? What is drug? Drug is a chemical molecules. I'll show you chemical molecules somewhere here. I show, yeah, like this. So this paracetamol, most of the people know paracetamol. The paracetamol drug, how can we represent it? Like this, we can represent CC bracket equal to 0. There is a significance meaning for each character. This, how can we identify such drugs? So generated, see, already we have so many chemical databases, Campbell, Moses, like that. We have lots of chemicals are available. Now, out of those many chemicals, which chemical work like a drug for a particular disease, we need to identify. For that, what we can do, there are two techniques. One technique is use existing drugs and test on the protein disease. Test on the protein. Protein means diseased protein. It is not normal protein, diseased protein. The diseased protein we can represent as 3D structure. Whatever the chemical structure is there or chemical is there, you can overlap on the top of the 3D proteins, diseased proteins, and how much effectively it is binding. The binding affinity score, one parameter is there. Based on that score, we will decide whether this chemical is effective work like drug or not. Okay. So for that, already existing chemical, so we will take it and test and bind on the top of disease protease. But this is a very time consuming process. Why? Because lots of chemicals are there to test on that. Even we required high computational powers systems. There is a tool like PyRX docking tool. In that tool, to do with 100 molecules, it is taking hours of hours time. So this is a time consuming process and another costly process also. Now, thus we can take the advantage of deep learning, generative adversarial networks. Okay, what we can do, we can build the model by using existing chemical space. We will take 2 lakhs or 3 lakhs chemical molecules. We will build the model. Next, we can generate novel molecules. That is one mechanism. We generate finite number of novel molecules. By using generative adversarial networks, we can do it. By using LSTMs also, we can do it. Okay. Some architecture, some simple architecture I given. I'll show that. We will complete this terminology. Next, after building that model, we can generate some molecules and we can fine tune the same model with existing commercial drugs also. So there are existing commercial drugs, right? For some diseases, we have already drugs. Okay, some HIV disease, some drugs are there. So some diseases, for some diseases, already existing drugs are there. We can fine tune our model by using transfer learning, and after that, it will generate very effective novel molecules. Very effective novel molecules. That is one more mechanism to generate novel molecule, drug molecules. Now, after generating the molecule, we need to validate it whether the generated molecule is proper chemical molecule or not. Maybe it is generated randomly some useless characters. So that we have to test it. There are various parameters to validate the generated molecules. Next, those generated molecules, finite number of generated molecules, we can build new deep learning models to test drug target interactions, or we use docking tools to test how effectively these novel generated molecules binding to the protease. Suppose if you take coronavirus protease, 3CL Pro. Okay, 3CL Pro is coronavirus protease. So 3CL Pro, we will put this novel generated molecules. We can dock on the top of the 3CL Pro. How it will give you some value, some dissociation constant value or binding affinity score value. If binding affinity have more negative value, it represents that this chemical molecule is effectively binding. Binding means it is effectively halting the translation process. That's the meaning. So other mechanism is by using deep or reinforcement learning, we can build the drug target interaction models instead of docking tools. That is another mechanism we have. So now, what are the terminology we use in drug discovery and design? One is molecule, a group of two or more atoms held together by chemical bond. This is molecule. So next, ligand is a molecule that binds to a site on the target protein. So these molecules, we can call it as ligands, which are effectively binding to the protein. Now inhibitors and a drug is a chemical substance. Obviously, it's a one type of ligand, typically known structure, which when administered to the living organism produces a biological effect. Now bind site, where the drug or ligand interacting with the protein, that position we can say binding site. The binding partner of the micromolecule is often referred to as ligand. So now take it, this is a ligand, this is a binding site. If it is effectively bind here, so it will bind here like this pictorial representation, that's it. Now how can we represent chemical molecule? We can represent, see, any chemical, it's a structured 2D structure chemical molecule, right? How can we represent in computer? So we can represent in this format, SMILES format. Usually we use this SMILES format, simplified molecular input line entry system. It look like this. We can convert these into numerical vectors, or we can convert these. There are huge research going on in drug discovery by using graph convolution neural networks. Each chemical molecule we can represent as graph, and each protein we can represent as graph, and we will pass to GCNN, and we will bind how effectively they are binding and so on. So lot of research is going on in this area. So SMILE alphabets, just go through if you are interested in this area. Okay. Now, generative model. How a generative model? Suppose there are three symbols in the chemical space. Just a simplicity, we took only three symbols, C1E. How can we represent C? 100. How can we represent 1010? How can we represent E means? So 001 like this. So this is LSTM. This is simple LSTM. Maybe it's a recursive nature. Okay, LSTM is a recurrent neural network, right? So it is a recursive nature. It will feed forward neural network again. So it will send the previous input, important input to the further steps or further time instances also. So if you give one character, first character, so it will generate some numerical vector. Based on the numerical vector, we will decide which character it is. And again, we will pass to next time instance, again next in time instance, and so on. Finally, it will generate some chemical molecules. These chemical molecules we can say novel drug molecules. So how can we validate it? These measures we use it, valid and unique molecule, novelty, all these we can test it. And by using commercial drugs, existing commercial drugs, we can fine tune the LSTM models or generative adversarial network models, and we can generate the novel drug molecules. Okay, so after generating novel drug molecules, okay, what are the techniques to decide which molecule is effectively binding to the virus protein, or what we can say diseased protein? So that PyRX tool is there, okay, to identify binding affinity and measure the and reported equilibrium dissociation constant KD value. We will calculate it based on the KD value. If KD value is less, greater binding affinity exists. So if there is a very less KD value, so we will decide, okay, this molecule is effectively binding to the drug. You see here, this is the drug. Just to take in 3D view, this is the drug, this is the ligand. Okay, so how it will bind? Here you see, it is binding here, it is binding here. Not only it will bind here, it will bind somewhere here also, somewhere here also, somewhere here also. So many positions it will bind it, but which position it is giving best binding affinity score, that we will need to identify. Again, very recent research, 2013 onwards, 2000 end of 2022-23, combination of these ligands, or we can say subgraphs of these ligands, group of subgraphs we are using to identify effective drug. Such research is going on. Okay, such research is going on. This is through PyRx. We can dock it on the top of protein. This is a protein. Next, another mechanism is we can build our own deep learning models to decide or to find the drug target interaction between drug target interaction is exist or not. Okay, so for that, what we can do, we required a pre-processed, we required a data set, pre-processed data set. In that data set, a protein is there, one ligand is there, and one label exists, whether there is a interaction between the protein and ligand. What we can do, we can pass the protein to the convolution layers, and we can pass ligands to the LSTM layers, and so on. This is one type of architecture. One type of, we can concatenate those outputs. Again, we will send to convolution, LSTM, and so on. Finally, we will build the model, and it will give, if you pass ligand and protein to the deep learning model, it will generate the relation between protein and ligand. There is a binding affinity or not, or there is a binding or not. So here it will generate binding affinity value. Here it will generate binding affinity. This is a regression problem. It will generate binding affinity value. Based on that, we will decide, okay, these ligands are effectively binding to the particular virus protease. Okay, so like that, we can generate few ligands, like our group of ligands, which will effectively work for particular disease, and we will send those generated and effectively interacted chemical molecules to the labs for clinical trials. Okay, what is the advantage with this means, we can build model within hours, very quickly within hours, and we can generate molecules, we can fine tune it, we will test it if there is a drug target interaction or is there or not, and how much drug target interaction is there, and so on. This we can do very quickly within hardly days. Okay, then we will send only few limited number of molecules to drug trials. They will do in the labs. Okay. In such a way, the deep learning or machine learning or data science helps to identify drugs candidates for particular diseases. So this is one best real-time example of conclusion portion. What data science? See, obviously, it's always confusion. What is data science, machine learning, deep learning? There is some interaction between all these. Okay, you take those techniques, the intersection techniques. We use those techniques to solve bioinformatics problems, leveraging its expertise in handling large-scale biological data. If you think a genome, human genome, it is 1 GB of a continuous string, right? To handle such huge files, okay, huge sequences, we required these techniques, okay, data science techniques. Now developing computational models, we can develop computational models for various applications, and extracting meaningful insights. Just we extracted one example like patterns which are effectively useful, and we can, in disease prediction, we can extract patterns which are due to which mutations, which type of disease occurring. Such meaningful insights we can extract by using data science. Now data science techniques such as data preprocessing, dimensionality reduction, feature selection help to extract meaningful patterns and reduce noise from these large data sets. So this point, pre-processing, this noise means in genome sequences there is a huge noise for the first time, continuous after generating in different versions. After regular intervals, they will update the sequence and they will enhance the sequence. Suppose if you take a noisy sequence, we need to check, is there any special character in the sequence, how to eliminate the special characters. If there is n, how to eliminate it, how there is a space, how to eliminate it. Such pre-processing we can do it by using data science. Now the integration of data science if you take it and with bioinformatics, has advanced to understand biological systems, provided insights into diseases, low cost drug design at rapid speed, and so on. So here we discussed only just three applications. First application is like signals predictions and the patterns which are placing crucial role to identify such signals. Second example is coronavirus evolutionary relationships, mutation rate, and translation rate related techniques. And another one is how to design a drug and how to perform docking and how to build drug target interaction models to identify the drug candidates for the particular diseases. So here I want to show you some protein data bank. Somewhere I shown, right, protein data bank. So we go to protein PDB, protein data bank. See, if you go through here, deposit, we can deposit proteins, we can search proteins, we can visualize proteins. Okay, visualize protein. Okay, so just if you go here, we can visualize it. How can a protein is visualized into 3D structure? It's a little slow, I hope. A lot of options are available here. The structure. So it will load related. Suppose this, you see, got this bat coronaviruses HKU4 is UDC. So the structure look like this. So this is the structure. So from here we can extract such structures, and PyRX tool also. So many other tools. PyMol is there to visualize the 3D structures. Okay, to visualize the 3D structures. So 3D structures looks like this. The molecule or chemical molecule bind somewhere. Okay, here on the top of this chemical structures. So here PDB, protein data bank. This is a primary database where we can store different proteins information. So if you go through documentation, basic search mechanisms, advanced search mechanisms are there, browse and visualize, analyze, different features are available. COVID-19 related information also stored here. So please go through it. If you have interest in bioinformatics, this is a very good source of data. And if you want to do particular research on specific areas, you need to search through literature. You need to identify the data sets. So then you go to PPT. Next, some references given here. That's it. That is the end of the presentation. So thank you very much for your patience, and I'm open for questions. Hello. Any questions from participants?
Participants, if you have any questions, please ask.
Yeah, from my side, yeah, I can understand the pain because this is completely new terminology for other research area people. But there is a high scope in this research area. The beginners who are going to start the research, this is also one open area where you can work and where you can solve real-time problems. So yeah, that's it. Richa ma'am, if there are no questions, can we wind up the session?
Sure, sir. We can wind up the session. We have got the material